Hey everybody, my name is Brady, and this is my second local expert project over RNA interference. So before we actually get into how the interference is caused, I just want to make sure that we're all on the same page here. So we have DNA. DNA is then transcribed into RNA. That RNA, using ribosomes, is then translated into proteins. Where RNA interference comes along, however, is we want to make sure that this protein is not being created, so we can effectively cause like gene silencing without actually augmenting DNA. So take double-stranded RNA that's identical or complementary to what we want on the RNA strand that we're trying to silence. Then you take this dicer enzyme, it actually slices it down the middle to create a single strand of RNA, which can then bind onto the antisense strand of the RNA that we're trying to silence. The Argonaut protein then takes that strand, combines it, and forms this thing called the RNA-induced silencing complex, which is then able to bind onto the RNA strand itself. And so one of two things can happen right here. So either that the binding is so good that the RISC or the RISC complex is actually able to cleave the RNA at those 2 points and then thereby degrading the RNA. Or if the bearing is not good enough, they're actually able to just stick onto the RNA itself. So when that happens, as the ribosome travels along the RNA to translate into proteins, it's actually not going to be able to. It will get like blocked by the RI by the RISC complex, and therefore we won't get a protein, causing gene silencing or gene knockout. So while RNA interference does have some like promising applications in the future, there are a couple of downsides that it has that we haven't really figured out yet. But onto the benefits, RNA interference is highly precise, which is very good for like potential research. Because if they want to target this one particular mRNA to see what'll happen, they're able to do so without actually potentially targeting anything else. It's also very good because it can be used to target a wide variety of genes, which has a lot of therapeutic applications. So we can target potential like cancer cells, their DNA, viral infections. We're actually able to like design it just particularly to make sure that they don't create the proteins they need in order to keep doing their harmful functions towards humans. It's also great for crops. Current plants are being treated with RNA like pesticides, where that once a pest consumes or tries to feed onto a plant that has this, it actually like cause birth defects onto their offspring to make sure they will not be able to reproduce and feed on those plants again. On the downside, however, we have to consider about potential off-target effects whenever we use RNA interference. So even though we're able to completely silence this one gene, who's to say that that gene does not regulate something else down the line with the protein that it creates? Along with that, it's also dose dependent, which is where knockdown and complete comes from. So not all are entirely effective once we're able to just like slap some RNA in there and call it a day. We actually consider just like, okay, so how much RNA do we actually need to produce and keep applying to it in order to actually make sure that it's completely silenced? There's also the problem of actually getting the RNA into the cell because, once you introduce foreign material into an immune system, it's going to attack it, obviously. So by the time that the RNA gets to the cell to cause interference, it could be completely degraded and entirely useless. So, saying all this out loud, it's very obvious that this technology and method can be compared to CRISPR, which we covered previously, that also covers over genome editing, gene silencing, whole 9 yards. The big thing, however, I want to point out is that CRISPR is more for DNA modification at the DNA level, meanwhile RNA interference is more about gene silencing at the post-translational level. So going back to this diagram I showed earlier on, CRISPR actually modifies DNA at the DNA level, meanwhile the RNA interference creates that RISC complex that goes onto the RNA and prevents protein translation that way. Like on a larger, bigger picture scale, CRISPR is more for permanent genetic modifications. Meanwhile, RNA interference can be considered more like one and done sort of things, where like once the RNA is in there, it'll prevent from creating the protein, and then we can see what effects those have later on. So for controls for RNA interference, it's kind of hard to describe because it's more to make sure that we're focusing on the outcome instead of the process. But there are still ways that we can test for this. So, like a positive control is to make sure that essentially 1 + 1 = 2. That once we introduce this RNA into the system, it's going to target this specific important function, and it should result in an outcome we can expect. So, it'll probably result in cell death if we are targeting something that important. Net control is to make sure that anything that we do to the cell is going to be as a result of the specificity of the RNA sequence. So, Once you introduce that to the cell, it should make sure that nothing actually happens to it. As for scribble non-targeting small interfering RNAs, it's to make sure that once this is introduced to the system, it shouldn't bind to anything. It's going to control whether or not there's actually off-target effects. Because if there are, then it could be, okay, maybe this RNA that we designed could interfere with something down the line, or it actually isn't going to target our gene in question. So with figure 1 right here, it actually shows that off-target effects can actually occur in organisms that you're not actually targeting. So there was so much similarity between honeybees and the actual pests on top right here. They had to go back and redesign the RNA vector and make sure that it's not actually killing the honeybees inadvertently when they treat the crops with this RNA pesticide, which I think is kind of cool. Not the fact that they're killing honeybees, but the fact that it's able to be so precise that they had to go back, find a different process to target or a different RNA sequence to target in order to make sure that it's specific enough to go against the pest and not the honeybee. This figure right here is actually trying to show how the pathway occurs when it's going through dividing cells. So the big thing right here is that they actually introduce extracellular vesicles that contains all of the small interfering RNAs as the cell divides, which once that happens, they get the double-stranded RNA to go into the cell. The virus actually replicate that into creating small interfering RNAs, introduces itself to the RISC complex, and is able to then modify the DNA at that level as it spreads downward into the daughter cells, which I thought was interesting. Double-stranded RNA or just RNA interference in general is very good for dividing cells because they're more likely to uptake that information, whereas CRISPR is better for like non-dividing cells because the genetic information is already there. So just another way that these two methods can differ. But it's also cool that they're just able to like introduce this genetic information, and there is a way using viruses to make sure that it actually gets in the cell without being degraded. Finally, going back on how I was saying that RNA interference is sort of dose dependent, this figure right here is actually showing that this protein, depending on how much small interfering RNA you actually introduce to the system, decreases sharply once you reach a certain concentration. It's just going to show that you need to actually have enough RNA to interfere with the actual processes, or just the fact that you need to have enough to make sure that once it's actually introduced to the cell, not all of it is degraded to cause an effect. So here are the sources and references that I used in order to actually create this presentation. If you have any questions, please shoot them to me and I'll be sure to get you an answer. And if I can't do that, I'll find somebody smarter to get you an answer. Thanks for watching, everybody.